Jimmy, this side, there's one of these.
There you go. Okay, so we'll talk biomonitoring then, so Colin asked Ginny and I to have a look into some of the possible techniques that you can use on your trip, and we're coming in fairly cold to the project, but we have been given some background information, so you're probably more clued up on this than we are at this stage, but as far as we understand it, the main aim of this field trip is to assess the consequences or the success really of river restoration, and that's using a variety of parameters. So as far as I understand it, you're going to be doing things like habitat surveying and the sort of physical measurements. Is that right? You're all right. Okay, so I'd strongly recommend you sort that sort of side of thing with Colin, and then we're here to deal with the biological side. So yeah, the idea is you're going to use a bunch of parameters to look at these different these different stretches that reaches a river that have been preserved in different ways. So this one was restored in 2009 to 2012, so it's got a kind of diversity of habitats. This one was more recently restored, and then there's a third reach which is classed as degraded, right? So that's the overall rationale for the project as far as I understand it, but as I say, it's not my project, and we're here to give you some of the nuts and bolts of the methodology. So the aim of this particular lecture is to give you a general introduction to biological monitoring or biomonitoring, and so it'll just be a methods introduction and something that we need to work on together, and also as part of the broader team when you get together, is exactly what you're going to do in terms of sampling plan, because we can give you the idea of what the kind of things you need to do, but you need to plan exactly how many samples you need to take. So what is biomonitoring? So it's the science of observing ecological conditions in a variety of aquatic habitats by examining the organisms that live there. So you can use biomonitoring, technically speaking, on any ecosystem, but here it's going to be in a aquatic context. And generally speaking, you would look at a community of organisms, and a community is a specific ecological term that means a group or association of different populations. So a population would generally be one species, so it's associations of different populations within the same geographical area in a particular time. So the classic thing that we think about is using invertebrates, which you've all done that, right? You've all had a go at monitoring benthic invertebrates. So there's different species, and that can tell us about the conditions. So you can use lots of different types of biota in order to biomonitor. These are the two that we'll be focusing on for this project: invertebrates and algae. But you can use fish, for example, but it's more tricky working with vertebrates. You need all kinds of permissions, and it's not very nice fishing either, not nice killing fish. So we won't be covering that. I don't know if there's an opportunity to do that when you're out there. That will be dealt with over there, if there is. Okay, so why do we want to biomonitor? So the biology is sensitive to environmental conditions. So by looking at the biology, we can get some sense of what the environmental conditions are. And one of the big advantages is if you take, for example, one water quality sample, that's just recording what's in that space at that particular time. If you've got organisms that have been living there over the space of days or months, they're integrating the conditions over that period of time. So they can arguably give you more of a integrated idea of what the environment is like. And then it also allows us, in this context, because we're trying to think about the success or otherwise of restoration, it allows you to measure a variety of biodiversity metrics. So you can measure species richness, for example, and you might expect that if your river has become more heterogeneous, that you'll have more habitats, and so you might have more species. So that might be one hypothesis going into this, that your restored reaches are more heterogeneous, which you can measure using your physical measurements, and then see whether that translates into more biological species richness. So this can give you a measure of so-called kind of ecological integrity, how sort of good or good a habitat is it providing for organisms. So one of the challenges when monitoring rivers is they're very heterogeneous, and that's why they're quite interesting to look at in terms of physical structures. So you might think of habitats like riffles and pools, for example. So there's heterogeneity, heterogeneity in a sort of vertical scale. So we've got the river bottom and all the substrates that are on the river bottom, and then we've got the water on the top. So what generally tends to happen is most of the biota that are sampled in a river will be associated with the bottom substrates, because that's the bit that is a little bit more stable. If you've got really slow flowing rivers though, you can get things that live within the water column. So plankton that live within the water column might develop in very slow flowing rivers. So if you think about the River Thames, it looks very turbid and dirty, and some of that colour will be due to the development of plankton which grow in the water column. Now the rivers that you're going to be looking at are presumably going to be a little bit more fast flowing, and in those kind of conditions, the very fast flows are not usually suitable for the development of floating organisms. So we'll come back to this in a minute about whether it's worth thinking about sampling plankton in this context. So first of all, thinking about your bottom substrates then, so also called benthos or benthic organisms that live within them, and because of the flows in the river it rearranges the materials in it and leads to different habitat structure that creates different conditions for the animals that live in there. So the types of animals can tell us about the habitat heterogeneity, and these are all probably what many of you are familiar with. We've looked at these certainly in Malham. We've sorted lots of these, and I know quite a few of you worked on these for your dissertation. So they're really easy to sample and visible to the naked eye, and usually dislodged from the riverbed by kick sampling or jab sampling, and we'll talk about that in a minute. What you're probably less familiar with, because I don't think we do a lot of work on this, is the types of algae that might live in rivers as well. So there are lots of different substrates that algae can grow on. So there might be mud surfaces, so you get these kind of biofilms which live literally on the top of the mud. This is a core sample that's been taken from the mud, and you can see the algae growing on the top there. Really commonly in rivers, you get rocks and cobbles or boulders which are covered in this kind of slime. So you can see that slimy surface there. That's actually quite a substantial biofilm. What you can also get is rocks that just feel a little bit slippery. You know, when you're out hiking and you kind of walk on a rock and they're often slippery in water, it's often because there's a biofilm of algae growing on them. So rocks are a good habitat for algae. Wood often gets, the wood that's been in there for a while gets algae associated with it. But you can also find algae free floating. This is a photo from, I think this is from Attenborough Ponds. And you can see there are these kind of twisted masses of filamentous green algae. So these are termed macroalgae because they're bigger than most algae. They might just be floating around. And you get other sorts of algae kind of living associated with them. So some of the characteristics of these algae, which are termed benthic algae or phytobenthos or periphyton, lots of different terms for them, are, once you look at that slime under a microscope, you realise there's a whole community going on that you can't see with the naked eye. So there's lots of microscopic organisms, and there's different ways of analysing these. Some are soft-bodied, and so you can kind of just see them under a microscope, but you can see there's also lots of debris and dirt kind of mixed in with them, so you have to think about how to get the samples prepped so that you can see them properly. What you can do as well is use the diatoms. So you've seen diatoms, haven't you, in some of your lab practicals? One of the advantages to that is you can digest away all of the organic material to leave the diatom shells behind because they're made of silica. And once you can focus in and see the shells, you can zoom in a lot better in terms of the magnification and look at identifications to species level. And once you can get identification to species level, that can give you a lot more sort of information about what's there and what habitats they prefer. So diatoms have been used a lot for water quality assessments. So this looks quite daunting. There's lots of different types of things to analyse, but you can make some decisions about how thoroughly you want to do this analysis. So, for example, you could count the number of filamentous blue-green algae to diatoms, and that will give you some idea of the types of community assemblage, or you can go further down and split further into sort of genus and species level, depending on the requirements. Okay, so your task really is to devise a suitable biomonitoring plan to assess river restoration. And what I've just shown you, this idea of looking at benthic invertebrates and periphyton or phytobenthus, is what most river sampling biomonitoring programs look at. Those are very common methods of looking at this. But one thing that we were kind of thinking through when we're thinking about planning a monitoring program here is that there are quite a few small backwaters and ponds probably in these areas that develop in the restored reaches, right? So when you've got restored reaches, you're leaving in the woody debris that can impede some of the water flow. You might get beavers creating dams. So probably you're going to have some very kind of calm, watery areas that might lead to the development of plankton floating in the water column. So the question is it worth sampling the phytoplankton? Because probably this is going to be quite a good indicator of the development of these kind of lentic, these non-flowing water areas. So this might be something worth considering. So do you want to consider sampling the plankton as well as the bottom organisms? It's quite straightforward to sample plankton, so it's almost kind of what we tend to do when we go out sampling is if you're out there anyway, sampling the benthos, it's as easy to take the samples, even if you don't end up counting them at the end of the day. So I would recommend you at least sample them and then think about whether to count them later. So the things that live floating in the water tend to be pretty small. So these are zooplankton, and they're probably just about visible to the naked eye. So they might be about 1 millimetre across. This is a water flea and a copepod. And usually you can just see them kind of flitting around in the water and swimming around. So they're relatively easy to sample from the water column. And as I say, these will only really develop when you've got fairly still water conditions. So they're like an indicator of still water conditions. So if you sample in the main stem of the river where you've got high flows, you might get none. If you sample in the slow flowing parts, you might get some, and that's an indicator of heterogeneity of flows maybe. And then this is an example of what you can get in terms of phytoplankton. So these are the plants, the algae that float around living in the water. These are smaller than the zooplankton, and they're eaten by the zooplankton. So you can't see them in terms of, they're not visible to the naked eye, but if the water's cloudy, it'll tend to suggest that you've got phytoplankton growing in there because it's caused by lots of algal growth. So let's think about how to sample these things. I'm going to run you through some of the sampling methods. And you've done benthic macroinvertebrate monitoring before, and they're very, very widely used. What we've done, Ginny's helped put the booklet together. Well, in fact, Ginny put the booklet together. Oh, and it's based on the US EPA protocols, but these are archived protocols, right? So I'm not sure how, whether they've been updated, but these were available, and they're a good start actually in terms of thinking about how to do this monitoring. They've been used since 1989, and they're known as rapid bioassessment protocols. I thought it'd be appropriate to use US methods, given that you're in the US. They've been kind of developed to cope with the types of substrates that you'll find in US rivers, so that's why I thought that would be the most appropriate one. And the way that they, one of the most widely used ones is a multi-habitat sampling procedure using a D-frame dip net. So we've got these things here. So there are standard dip nets that you use. So standard dip nets that you use in the US, which are particular dimensions. So this is all given in the protocol. These are smaller than the standard ones. So you could decide you're going to use these smaller ones, but it has to be a standardised net size. You just decide on that. Or we could get hold of some of these. I don't know how this is going to work, so it's something to discuss when Colin gets back and with the technical staff who will be helping to kit you out, whether we take these over, which seems like a bit of a hassle because I think they're pretty cheap, or whether they've got a bunch of these in the US. So yeah, one for you to talk about with Colin. So we've got a protocol booklet that we're going to circulate to you, so you can have a look through some of those protocols. And also, all this stuff's on the web if you want to look at. There's really, really detailed methods, and it's all been edited down to the things that we think are more pertinent. So as I say, there's this multi-habitat sampling technique for benthic macroinvertebrates, and the idea is that because you've got this spatial heterogeneity in habitats, so you might have kind of a woody area, or a bit where there's plants, or a bit where there's riffles, a bit where there's pools. You start off by doing your physical habitat survey, and then you quantitatively sample the different habitats relative to the proportions in which they're available. So yeah, so that's what you're trying to do, get a quantitative representation. And so you need 20 samples per reach. So one of the questions I've got, which I don't know about, and I don't know if you know the answer to this, Liam, at this stage, but so this is designed, the 20 jab thing is designed for a reach which is about 200 meters in length. Now, if this entire, there's no scales on any of those maps we've been given, so I can't work out how long they are. So if it turns out that your entire reach that you're looking at for each of those bits is, I don't know, 400 meters, then you might want to rethink whether this is an appropriate way to do it, or whether you do single habitat sampling. So what you could say is, right, along the whole reach, we're going to do, I don't know, 10 samples of rocks, 10 samples in riffles, 10 samples in pools, 10 samples in bankside vegetation, or whatever. So that might be the way that you decide it's more appropriate to compare. But yeah, so that's a discussion that needs to be had. So yeah, you take your sample, which is either a kick sample or a jab sample, combine them and preserve in methanol. And then back to the lab, sorting through the material that's been collected, and then identification. So I'm not going to talk much about identification today. So yeah, so the question that you need to discuss among yourselves is a combined sample suitable for your research question, or would it be better to sample individual habitats? Do you have any thoughts yet, Liam, on the sort of scale of these reaches in terms of what?
Next week we've got a meeting on Wednesday morning.
Great.
Which
Okay.
Is, the wrong way around to do this, but
yeah. Well, but at least you know kind of what to discuss and how to get these sorted out. So just to give you this is an idea of the main habitat types, the invertebrate habitat types: cobbles, snags, which is woody debris, where you do a jab sample, vegetated banks, submerged macrophytes, and fine sediments. So you would sample those in the appropriate way. So the main types of samples are jab samples, which is forcefully thrusting the net into a productive habitat for a linear distance of 0.5 meters. Did you want to demonstrate that, Ginny?
Maybe after the echoes.
And then a kick sample, which you've all done right. So the idea is you stand downstream, so you're kicking. Sorry, you stand upstream, so it's washed downstream into your net. Okay, in terms of benthic algae monitoring, it's a little bit different because you're basically monitoring. You're picking bits of slime out the river, right? So it's not the same type of technique. So benthic algae are essentially the primary producers. They're at the base of many stream food webs and sensitive to water quality. So a lot of the protocols that were developed later than the macroinvertebrate types of survey methods, but they're now really widely used for biomonitoring. So diatoms are used a lot for biomonitoring in the UK, for example. There is a multi-habitat sampling periphyton method, which again integrates these different habitats. So again, you need your habitat survey to work out the lie of the land in the river and what types of habitats you're going to be looking at. And you determine the percentage coverage of the substrate types, so the relative abundance of macrophytes, macroscopic filamentous algae, periphyton, and other biota. And then you get your composite sample from all the different substrates. So you only need a small amount of sample, about 5 mils or less, because these are microscopic organisms. You don't need vast amounts of stuff. And so you combine them all into one common container. And then you preserve it using Lugol's iodine. So you're going to need to think about taking some preservative along with you, or getting some there. If you want to do a chlorophyll analysis, you need to subsample first because the Lugol's iodine will wreck the chlorophyll. And then they're identified in the laboratory, which sounds very simple, but obviously there's a fair few taxonomic hurdles to overcome first. So again, is multi-habitat sampling the most appropriate way to look at the periphyton, or do you want to think about doing some single habitat sampling? So, for example, some of the diatom methods for monitoring in the UK tend to look at, if you want to understand water quality, you can take just rock samples and compare in different parts of the river, and that will tell you about water quality. But your research question is a bit more about the kind of habitat heterogeneity and how that's affecting the biota. So I'm kind of thinking you need to be looking at these different habitat types to answer this question about heterogeneity. So there's some info on how to sample these different substrates. So on gravel pebbles, you basically get your pebble and toothbrush it off into a jar. You're rinsing off the pebble. So just remember, most of the algae material is going to be on the upper surface of these rocks because they're photosynthesizers. They need light, so they're unlikely to be growing underneath the rock. So usually you would monitor the top of the rocks. So if you've got plants, for example, you can put some plant in a jar, shake it up, and that'll remove the algae associated with the moss or the plants. Large substrates. So this is the idea of getting a PVC. You're basically trying to put something on the rock that you can toothbrush and then kind of rinse into it. We've used things like, actually, yeah, you can scrape off as well with, I don't know, a razor blade or something. We've used a technique piloted by Ian, which was a mouse mat with a hole cut into it, so you know what the sort of hole is that you're removing, and then you remove it with a pipette. And then loose sediments, yeah, so you basically get a Petri dish and then stick a spatula under it, so you really just want the very surface biofilm. So it's quite hard to standardize the amount that you're sampling. So yeah, so I put these methods as semi-quantitative. And then it's microscopy to identify them. So because we've got like an algal soup that we get, you've got to have a kind of a cell that you put it in, or a sedimentation cell where it goes down to the bottom, and then you look at it under an inverted microscope. So So yeah, you need to discuss what sort of samples you're going to be collecting, and then therefore what kind of microscopes and counting material is necessary in order to quantify them. If you want to look at just the diatoms, you take your algal soup and you digest away the organic matter, usually using peroxide or something, and you leave just the diatom pustules behind, just the silica material, and then you can mount them on slides. So how quantitative do you need your periphyton analysis to be? So what would be the advantage of being able to quantify the amount of algae in the river? I mean, what will it tell you? If one lake's got loads of algae in it and another's got less algae, what might that suggest?
Different nutrient levels.
Yeah, so it's suggesting there's some sort of difference in productivity, or yeah, the amount of nutrients, the baseline nutrients. So by doing that multi-habitat survey, because you're getting more or less the same amount of material from these different parts of the river, I reckon it would give a kind of ballpark estimate of which is more productive and which is less productive. But if you want to be, if you want a really good quantitative comparison, what I would do is to get some rocks to measure the area that you're scraping for periphyton, and maybe compare between the rocks, the actual amount of algae that you're removing. Depends how much detail you want to go into it, depends how much time you've got, how you want to allocate your workloads. So, you know, there's different levels of detail we can go into for this. So one way of not having to count the algae, but still getting an estimate of biomass, is to do chlorophyll or carotenoid pigments. So you've done the chlorophyll analysis, right? When we were in Malham, we used chlorophyll to look at phytoplankton. So what you can do is you can take the algal gloop, and what you need to try to do is subsample it so it doesn't get preserved with the Lugol's iodine, freeze it, and then you concentrate it up onto a filter paper, and then you can fold it in half and freeze it. So you need to try and keep it out of the light basically and get it as cold and frozen as soon as you can. And then you can analyse by spectrophotometry, which will give you an idea of the chlorophyll A, or high performance liquid chromatography, which will give you an idea of the composition. So there's different pigments in the different algal groups. So we can do that back here if you wanted to preserve that. So the suggestion would be really that if you're going to go out and monitor these biofilms, you might as well take some of that sample and freeze it to analyse it later, because it's pretty easy to do. So another question to discuss among yourselves is, you know, do you need quantitative samples? Do you want to try and estimate quantity of phytobenthos between your different river reaches? And if you do, then you probably need to be sort of standardising how you take your samples. So the heterogeneity of the rivers and the substrates really creates quite a few methodological challenges for collecting phytobenthos. But the information you can get from phytobenthos is really quite informative. So that's thinking about how to monitor your bottom substrates. The plankton, as I say, this is something that is not routinely collected in fairly fast flowing rivers, but I think this might be quite interesting, and it's also pretty easy to collect. So what you would tend to do to collect zooplankton, because they're kind of these very small organisms that are not, there's not a lot of them in the water. So this is a zooplankton net. So what you tend to do in a deep water body is drag that net through the water body. Yeah, so normally you can drag it vertically, and then you can see how much you're basically filtering that water column and concentrating up all the organisms. Otherwise, there's just, you know, if you just take a water sample, there'll probably be like 2 organisms in it. That's concentrating it up. The problem you've got in a river, of course, is it's not deep enough to use something like that. And also, if you want to do it, you could tow in the river, but that's semi-quantitative, or well, it's qualitative really. So what you could do, I think, is so yeah, you basically take a known sample volume of water, concentrate it up using a mesh, and then preserve it using methanol sucrose, and then identify it. So what we could do is, in Attenborough ponds, because they're very shallow, what we've tended to do is use this sort of device. And the idea is, if you lower it into the water column, it will integrate all the depths of the water column. So you don't go right down to the sediment, because you don't want that. You're trying to get the water column. Bung it, so you put it in the top.
Oh,
Yeah. So and then it should suction it up, and then you can pull it up, and then you can stick your hand on the bottom too. And then bung it in a bucket, and then keep doing that till you've got, so your bucket, you'll measure how much you've collected. So usually 3 of those is about 10 litres. Yeah. And then you can filter that through your net. And then it's really, again, any water body that you're working in, you've kind of just got to have a look and see how many animals you're collecting. So, all right, I might need to collect a bit more. Yeah, so I don't know, 20 litres might be all right. Depends on the site. So I would expect you'll get very few of these animals in the fast flowing bits, but quite a lot of them in the backwaters and the sort of lentic parts of the river. So I think it'd be quite informative to look for zooplankton. So the question is, how much water would be required? We have no idea. And you won't have any idea until you get out there and look at it. So, you know, that's the nature of research, really. And phytoplankton. So how would you sample for phytoplankton? You can see here there's some examples of waters that this has got a lot of phytoplankton in it, so it's coloured. This has got less phytoplankton. And it's simply a matter of you sample the water, preserve it, and then you can look at it under a microscope. So you don't have to concentrate up or anything, usually. So you can use an inverted microscope or a sedimentation cell and to look at that. So again, how much water would be required? Not sure, because what you do is you put it into your sedimentation cell, leave it to settle. So often 10 mils is plenty in Attenborough ponds, which is fairly disgusting in terms of water quality. If you've got a less productive system, you might need to have 50 mils that you sediment out. So yeah, you might want to collect, I don't know, 50 to 100 mils of water and preserve it, so you've got plenty of sample to play with. You won't know till you look at it under the microscope. And then the other question is, where will you take the samples? So we don't expect that there'll be much plankton development in the fast flowing parts of the river, but you've got to kind of show that there's some, you know, showing habitat heterogeneity is what this is all about. So you might want to sample some fast flowing bits, some slow flowing bits to get an idea of how that varies. So yeah, as I say, the methodology is not well developed. This is more commonly used in lakes, but this is the researchy bit of it, the sort of quite interesting bit of it. And then, of course, you can do chlorophyll analysis. So we have these filtration units that you can use in the field. So you can get a known quantity, get a bucket of water, again, and pump it through your filter. If you measure how much you've filtered, probably a bucket's going to be too much. Yeah. But I don't know, a litre or 2 litres, something like that. Filter it through and it'll go green. So you just want some nice green colour on here. This has basically removed all the algae from the water. It's quite, one of the good byproducts of this is usually, if you've got one of these filters, they've got a 0.4 micron pore size, which means you can probably drink the water that goes through it because you've filtered out all the bacteria and stuff. So it's quite good if you're camping and stuff, because you can, that's all those things that you buy in the camp shops, that's all they are. They're just filters that remove the bacteria and stuff. Although if you all come back with Jardy, don't say that I told you to do that. So, yes, the volume, you need to note it down, and then you can extract the chlorophyll and work out the amount of phytoplankton in the rivers. So here's the other question, do you want to take some of these filtration kits so that you can get some easy estimates of phytoplankton biomass? So there's a bunch of different sort of techniques that you can potentially use there. And yeah, some of them are pretty straightforward and some of them are a bit of a hassle. So you need to work out essentially how many other tasks you've got to do, how many people there are, and how you're going to split up the sort of tasks among you. Yeah, they're just pictures of periphyton. So Ginny's drafted out this booklet.
Oh, a couple of copies here.
Yeah, so I'm going to circulate that by email, and I'll circulate the Echo lecture.
Sorry. Yeah, so it's basically just a slightly more in-depth version, but very much from the US EPA guidelines for either single habitat surveys if you decide to go down that route, or multi-habitat, particularly for the inverts and the macroinverts, and then obviously also for periphyton, and then the more traditional methods for the phytoplankton, the zooplankton, and like algal biomass, chlorophyll. Pretty much, yeah, again, phytoplankton. How many of you are going?
12 students.
Yeah, so there's quite a lot of people actually, yeah, and because those reaches don't look that large, so I think you could probably do something quite nice in terms of single habitat surveys.
And so obviously the equipment, I pretty much just list, because I wasn't sure whether you might like divvy up, like I'm going to do periphyton, I'm going to do the jab or kit sampling. So yeah, a lot of it is repeated, but I just thought then at least you know what you need if you discreetly kind of separate into smaller groups.
But once you've decided on this, obviously there's going to need to be a little bit of a lead time in terms of lab kit, because if you need filtration rigs, filter papers, that kind of stuff, those are really easy to take. They're light and stuff. Nets are a real pain, but I'm assuming there'll be some out there. I don't know.
That's my assumption, yeah.
Yeah. Okay. Because that.
Lots of different teams are already out there.
Yeah. Yeah. So it's
And I mean, have they already got this? Yeah, I don't know. Maybe they've already got a partial sampling plan.
Yeah.
But I think it's good for you to come up with between you your own kind of idea of if there are lots of different habitats, what would I,
So most of the kit is the same for single or multi-habitat, so that's something that, I think maybe when you've done actually your habitat surveys, you could even decide after that, because it's so linked to the habitat survey, what is appropriate, so. But yeah, the other thing that you need to think about as well, actually more for you actually, Liam, when you talk to Colin, is things like the preservatives like methanol and lugols. You can't fly them over, so they need to get them ordered over there. So, and sample bottles as well.
I was going to say, is that easier to, so for sample bottles there, I just kind of picked numbers from what I would probably take with me if I was off in the field. So I don't know whether that's the ideal volume to go for, or but I don't know whether that's something to take or just get there really. Buckets and stuff like that, you don't want to fly over with a bucket.
So normally when you're setting up something like this, there's quite a defined question. You're comparing these 3 river reaches, but in terms of the biological monitoring, it's nice to have some sort of hypotheses going into it, right? So, I mean, I guess I would hypothesize that the restored river reach is going to be more diverse in terms of biota. And I don't know what to expect actually in terms of productivity. I would guess it might be more productive because there's more connectivity with the terrestrial ecosystem. Obviously there's some reading you could do around this to help to inform those hypotheses, but if you've got a good hypothesis, that helps to decide what sort of sampling you want to do, because if you are interested in diversity, it's no good just measuring chlorophyll to look at diversity. So, yeah. In terms of kind of restoring these rivers back into kind of, I suppose, slightly messy ecosystems where you've got kind of ponding and these different types of areas, it's quite interesting really that there's classically no phytoplankton sampling or things done in these kinds of rivers, and so I think this is actually the more research wise, the more interesting research question, because you wouldn't expect to normally sample these things, but I think it's just because virtually all the rivers that get worked on are so degraded and not really what they would have been like in a natural state. And as far as I can understand, this is more of this is probably what the rivers in the UK would have looked like in well, not really that sort of forest, but 500, 600 years ago, before all these areas got drained, it's probably what they would have looked like. So it's quite interesting to think about, whether you want to measure different parameters. What other things will you be measuring, Liam? Are you doing GPS survey, or are you doing some levelling? And are you doing some what's it called? Are you doing a proper survey?
Yeah, I think
Yeah.
I think we use those questions. That was important because we know how to do it from having done it in America.
Yeah. Okay. So what did you do? Can you remember?
Measured like cross sections across the route of the river.
Okay.
That was what we did on the trip, and I think that was why Colin wanted us to go, because we already knew the protocol for that.
Great.
So we could find it.
So that'll be really good. And then in terms of, you've done habitat mapping, right?
We did a little bit in like largely debris, sort of like streams, but it was this. We went to like a eco centre, and the lady who ran it had a very like her own method that she was using, so it wasn't sort of like anything like this.
Yeah. Okay. So I think it'll be quite important if you're going to evaluate, the consequences of this restoration, to make sure you've got good habitat maps. Which it'll be easy enough if you're doing the sort of mapping anyway. You're doing the cross-sectional areas, and just to tie that in with, the mapping would be.
Yeah, so I
habitat
incorporate that
features.
in the methods, because obviously if you do go down for these kind of habitat assessments, obviously you need to know what percentage coverage you've got of each. So
Yeah. So
like
substrate mapping.
sketch and stuff, and then direction of flow, blah blah.
I'll be really interested to see how much kind of productive algal growth you get, actually, because I just don't know, and I imagine there's going to be quite a bit of woody debris in there, but I don't know how old it's going to be. So is it started to decompose yet?
So that first site, which was restored in 2009, some of the pictures that Colin sent through, it does show like July 2009, July 2010. I just took.
Oh yeah.
So you can see that already quite quickly. So that's the same tree. So you can see that already, like 6 months later, a couple of years later, 2 years later, it does change quite rapidly. So some of it might still be quite young. But I've got no idea what the flow of these rivers is like at this time of year. I don't know how big they are, but it doesn't look too big.
The stuff you were saying we could bring back to the UK. Most of us have graduated and gone.
Yeah.
So is he just going to palm that off for someone else to look at, or?
So that's what needs to be clarified, because I don't know who's doing the counting. So some of the benthic invertebrate counts are fairly straightforward and can be done with fairly simple microscope equipment. They could be done out there. I don't know what algae counting equipment there is, and I don't know if there's diet and prep equipment out there. I presume not, and that would need to be done later. So I think, but there's some students over in Oregon who I think are also associated with this project who might be able to do some of those counts. So so I don't know. but you're asking the right question. This is we're planning a project. We need to know what's going to happen further down the line. So, I don't know. Tom can do them all because he's here for a masters.
Anyway, you can bring them back. Take an
Yeah.
extra suitcase.
Yeah, take it.
Yeah, trust me, there was a time I had to do it. 3 suitcases.
Yeah. Yeah, so that's the plan so far.
Are you excited
That's-
to do some biomonitoring?
That sounds way more interesting than measuring cross sections all day. It's
Yeah,
like.
I might try it without some grapes.
Hmm. Are you doing any flow measurements as well? Is that right? Okay.
Yeah, Colin hasn't not told us anything in regards to what we're doing.
Right. Okay.
The other one was flying out, that helps.
All right. Okay. What time of the year is it?
July.
Okay.
End of July.
End of
How long
July.
are you out
Yeah.
there then for doing
10 days, I think.
10 days. Okay.
Hmm.
11 days, something like that.
Yeah. Because those reaches don't look that big, so it's logistically possible for sure to get 20 samples minimum from each stretch. So
Especially if you divide it into groups of 3 and you reach
Yeah.
10. Yeah. We could probably should have figured out from Google Earth how large they were. No. So
Right.
you're meeting Colin next week?
mm,
It's going to be a long meeting.
All together or just
No, just
you
meeting.
two? Yeah.
Yeah, it might be an idea to hold these as well.
Yeah, when?